Good morning, everyone, and welcome to the wrestlers class. Thank you for coming on this beautiful April sunny day. And we have an exciting speaker to share with you today. This is Earth Day, right? Among other things. So it's an appropriate topic for today. So let's get started with a verse of scripture that I want to share with you. This is from Romans 9:20 to 22, and it just says, it's a very familiar passage. But who are you, a human being, to talk back to God? Shall what is formed say to the one who formed it, why did you make me like this? Does not the potter have the right to make out of the same lump of clay some pottery for special purposes and some for common use? And that's the word of the Lord. Let's pray. And Lord, we do thank you for this day and for this opportunity to have some fellowship among ourselves, with each other, and to challenge ourselves to wrestle with questions that lead us to thinking about our faith and our relationship to you and to nature. We pray that you'll guide us and give us wisdom, guide our speaker, and we thank you for this fellowship. We recognize the presence of your Spirit among us. We pray in Jesus' name. Amen. So our speaker today is Dr. Seymour Garte. He has a PhD in biochemistry, and he is a professor of public health and environmental health services at 3 universities. And he recently retired from a senior administrative position at NIH, the National Institute of Health. Dr. Garte is a lay leader of Rockville United Methodist Church. He's the vice president of the Washington, D.C. chapter of the American Scientific Affiliation, and he has recently been hired as the editor-in-chief of God and Nature, which is an ASA's online magazine. So you're welcome to look at that. It's online at asa3.org and /God and Nature, I guess. And that would be, it's a collection of essays. It has art poetry and various stories, fascinating literature, and our speaker is the editor of that now. But his primary interest is in biology, evolution. As you know, David Sechrist was here earlier in this series and talked about some of the questions around genetics and genomics. So we're picking up on that same biological theme. It just keeps coming back. And Seymour Garte is going to share with us about the teleological biochemistry of evolution. So, Cy, take it away.
Thank you. Wait, hold it. I don't know what to say yet. Thank you, Paul, and especially thank you for inviting me here. I have been to this class before. I don't really remember. Do you remember when, Kat? I don't remember. It was a few years ago, and mostly what I remember is the trouble I had finding the building. It was one of those cold winter days, and I was wandering around. But this time I had much less trouble. So I'm glad to be here, and I'm going to talk about something that's a tad controversial. By that I mean it's controversial within biology, as you'll see in my first few slides. And it deals with what I feel is a fairly important topic, and that is very relevant to Christian faith. It's the topic of purpose. Paul asked me to choose a verse, and the verse that he read has the word purpose in it. Not probably in exactly the same context I'm going to talk about, but I still thought it was of interest. And all of this, everything I'm going to say, relates to a question that many people raise in Christian apologetics and debates with others. Is there a purpose to life? Is there a purpose to us, to the universe, to everything? And you've probably heard it said, it's been said by many people, that evolution is a purposeless process. That we have accidental mutations in DNA which occur by chance, and then there's a natural selection process which determines which individuals survive and which don't. But there's no end purpose to any of the actual scientific processes of evolution. And I challenge that view. I'm not the only one. There are quite a few people who challenge that view. I see a lot of purpose in evolution. I see a direction to evolution, and that's becoming actually fairly mainstream. A very well-known evolutionary scientist in Great Britain named Simon Conway Morris has led a research activity in what's called convergence. It turns out that evolution cannot do anything. There's a famous quote by Stephen Jay Gould which says that if you wound the tape of evolution back to the beginning and start all over again, you get something completely different, because everything is accidental, everything can happen. We only got one particular set of creatures. We could have gotten completely different sets of creatures. But that's probably not true. In fact, it's almost definitely not true. And the reason we know that is because the same things keep coming up in evolution: wings, eyes, intelligence, in different phylogenies, all from invertebrates, vertebrates, insects, mammals. You keep getting the same features, and this is called convergence. And what it seems to indicate is that there is a pattern in evolution that leads in particular directions. Very interesting. So I don't know much about animals. I'm actually a biochemist, but I could sometimes pretend to be a biologist, but I really know very little. Don't ask me what tree that is. I have no idea. I can barely tell a squirrel from a rabbit. I'm not that kind of biologist. I was born in Brooklyn, and we had squirrels. That's about it. My interest is in biochemistry, and that's where my PhD is, and that's what I've always been fascinated in, is what do we know about evolution at a molecular level? And what's interesting is this is very rarely discussed, even in the scientific literature. Evolution, as we'll see, turns out to be an extremely intricate biochemical process. Let's take a look at some of the slides before I go further. The first thing we want to talk about is, what do I mean by teleology? And that's itself a complex issue. Teleology, of course, is a philosophical subject, and my knowledge of philosophy is about equal to my knowledge of zoology. But I've done a little reading, and I've come up with a few things. So there's two ways to talk about teleology. Teleology is an Aristotelian term. There's a much newer term called teleonomy. I don't remember who invented it, but it was really made famous by Ernst Mayr, who was a giant in evolutionary biology. And it refers to the kind of teleology that we see in biology then. And let's see what we mean by that. Okay, teleology itself is the philosophical attempt to describe things in terms of their apparent purpose or directive principle or goal. So, you got in your car or walked this morning to come here. Okay, so you had a purpose, and you knew what that purpose was, and therefore you directed your actions to achieve that purpose, which was to arrive at this class. Me too. That's what's called a teleological, I think it's called the formal cause. I don't remember all the terms, but maybe some of you know them. So the question is that what we're talking about when we talk about purpose in biology? So in other words, does the giraffe look up at the trees and say, I think I need a longer neck when we get a longer neck? We already know that's not what happens. Okay. That was the old view. That's been, thrown out. So we're not talking about that. Another definition is a philosophical interpretation of natural phenomenon as purpose or design. I'll do that for a moment. And final causes, that's what I meant. The idea of teleology is it's a final cause, meaning you wanted to come here. And now you did all the actions necessary to come here. Now, teleonomy was used, since we know that giraffes didn't, deliberately extend their necks, that kind of teleology doesn't work in nature in biology. We kind of know that. But there is a sense of purpose because, in fact, when a rabbit sees a fox, it runs away. It has a purpose, which is to survive. The fox chases the rabbit. Its purpose is to eat or bring the food to its kids. So everybody has purpose, not just us. We clearly have purposes, but all animals have purposes, even bacteria. If you look at them in populations, act with purpose, which I'll get to at the end of the talk. But it's a different kind of teleology, and it's called teleonomy, and that describes a process that owes its goal-directedness, so it still has a goal. Okay, there's still a goal, to eat, to survive, to run away, whatever, but that is due to the operation of a program. Now, what does that mean in general? I'm not sure, but in biology it's great because we can easily identify that program. The program is the DNA, the genes, the metabolism, the mechanism of life, biological mechanisms. And we know that DNA is a program, so that fits well. But the idea of purpose, as I said at the beginning, is really controversial in biology, and many people don't like it. Especially nowadays, after the rise of new atheism, it's become very controversial. So Jerry Coyne, who is, some of you may be familiar with him, he is a fairly militant, atheistic evolutionary biologist, says this: he doesn't like the idea of purpose. It's a purely naturalistic process, blind material process, which acts according to the laws of physics. It has no mind directing it. I don't agree with any of that. First of all, biology doesn't work according to the laws of physics. It doesn't disobey the laws of physics, but it has a lot of other laws that are not found in physics. But I don't, if there's any physicists here, we can fight about that later. But anyway, it's not just physics, and I don't believe that it's blind, and I do think there's a mind directing it. Actually, I think several minds, but we'll get to that. And here are some other quotes of people who don't like the idea of teleology. Evolutionary theory did away with teleology, and that is that. I love that quote because it's very, very clear that we know the truth. Which, by the way, in science, if you ever hear anyone say, this is the truth, we're done, that is that, you know they're wrong. That's a definitive statement that I will, and that's probably wrong too. Definite regress. But this is a very important quote, the first one. Teleology is like a mistress to a biologist. He cannot live without her, but he's unwilling to be seen with her in public. That's J.B.S. Haldane, one of the great evolutionary biologists, and that's true. It's almost impossible to speak about biology without saying somebody did something for a purpose. A turtle came to the shore to lay her eggs, is the famous one. There are many people who say, no, you should never say that. You should say, the turtle came ashore and laid her eggs. There was no purpose at all involved. Okay. It's hard to avoid this. In every area of research, biologists are hard-pressed to find language that does not impute purposelessness to living forms. And here is the key issue. Ernst Mayr was very troubled by this whole thing. In his book, A New Philosophy of Biology, he devoted a chapter to this. And what he says in this third quote here is that you can say those words, the turtle came ashore to lay her eggs, and that does not impugn that there's any conflict with a purely physico-chemical causality, he says. Fine. So he's saying it's kind of silly to say, to forbid saying the turtle came ashore to lay her eggs. I think that's silly. And by saying that, you're not saying that it's some mystical, divine cause. God didn't, tell the turtle, thou shalt go ashore and lay thy eggs, or anything like that. So Mayr was trying to sort of, smooth this over, which he was only partially successful at. But in fact, not completely, because the truth is that there really is teleology in biology. Really, as I said, it's obvious. And no less than Daniel Dennett, who was one of the four horsemen of the New Atheism, agrees, which I find interesting. And by the way, Richard Dawkins, another one of the main atheists, does not agree with this statement, but he does agree that evolution has a direction. So it depends who you talk to. But I think Dennett's point, and this is only a small part of the whole quote, is that the biosphere is utterly saturated with design, with purpose, and with reasons. Yes, of course, his view is that those all come from natural selection. They have nothing to do with a God. But that's okay, because that's the first step. And I'm going to mostly be focusing on that first part of this point to demonstrate. Now, Dennett is a philosopher, not a biologist, and he just simply wants to say, look, I think he's a very smart guy, by the way. It's really silly to say things that are blatantly untrue. Let's admit that there is purpose and design, even, in biology. And then we can see where it comes from, and his answer, of course, as I said, is the evolutionary naturalistic process. Dennis Noble, one of the great, still living evolutionary biologists, and of course does not agree. I don't believe he's a Christian, but he is very open-minded about evolution. And of course there's Ernst Mayr with his correct comment that natural selection does not have a future goal in mind, and that's true. I may actually skip this. Wait, something is out of order. Sorry about that. Oh, I see. Okay. No, sorry. So, when we talk about purpose, I mentioned at the very beginning that even bacteria have purpose. Let's take a look. Now, some of you may have, does this thing look familiar at all to anybody? There's a very popular YouTube video. I hope it plays. Let's see what happens. I'm not positive. Oh, probably not.
No, I think you have to lift your mouse and click on the blue put your cursor on the mouse, not your finger.
Oh, this is not the touch.
Yes, now click. And if there's a hyperlink in there, it should pull it up.
It should be.
Push it twice.
Double click. Give it a second. It's not critical if we can't see this. Oh, something's happening.
Yeah, wait.
Thank you.
Is there audio with it?
There is, but it's not necessary. It's not important. What this should show, I think
Click on that little yellow thing down there.
The small printer.
yeah, processing request. Okay, we'll see what happens. What this is, I'll just explain what the experiment is. And if you just put in something like bacteria growing on a dish, it'll show up on YouTube. People took bacteria, put them on a plate, and they had antibiotic in increasing concentrations in different parts of the plate. Okay, let's see what happens. Yeah. And they wanted to see, would the bacteria be able to move into regions which had increasing levels of antibiotic, which kills bacteria. And that is not it. Oh, it's an ad, okay. Skip here. Scooby don't have the audio, actually. I should not skip that.
Skip that.
This is the first time in my life I've ever shown a video during a talk. I'm kind of old, so this is new.
You can do most anything with technology, but not in 5 minutes.
Technology has a purpose.
Yeah, that's what if you plug in Kishoni Lab and evolution of bacteria,
I don't wanna-
this is a very, very popular video. If it shows. Let's see, wait.
Yeah, the
Oh,
bar,
yeah.
yeah.
The bar food.
Okay, here we go. Right. So that's a very large agar plate. It's like 4 feet long and a couple of feet wide. And in the agar plate, there are different sections. And on the extreme left and the extreme right, there's nothing. But as you see 0, but as you go towards the middle, they put in a dose of of an antibiotic, and they make 10 times as much, then 100 times as much, and 1,000 times as much in the center. So no bacteria will grow if you just put them into those high concentration areas. But they started by putting them into the 0 on the edges. Okay, and there they are growing and filling up those 0 edges, and then they try to move. And then one colony with the arrow actually mutated and is able to live in 1% antibiotic. And now other colonies are also, on both sides, mutating. Each colony is one bacteria cell which had the mutation to resist the antibiotic. So then they fill up the 1, and now they start moving into the 10. Again, another mutation. Actually, each one is a separate mutation, as we'll see. And they fill up the 10, and then they move into the 100, and they keep going. And every time you see a new colony which then expands, that's one bacteria out of millions, which has undergone a mutation that allows it to so-called evolve. Creation's called this microevolution, but it's still evolution. And eventually, they even get into the 1,000-fold. And that is a demonstration to me of 2 things. First of all, it's the kind of thing that actually a lot of biologists do.
Stop the video, please.
Oh, we don't want to do that. Yeah, let's go to the next one. Okay. Let's see.
Do you want to go to the slideshow again?
Close the whole thing.
Yeah, do that right next up there.
Just close,
Yeah,
yeah.
and now, I can't read it.
Oh, it's in French. No, it's in we launch now. Oh,
No,
no.
because we want to go to the slideshow.
All right.
Now you can arrow forward to the next slide.
Got it. Well, sorry that you're all the guinea pigs for my venture into technology.
Yeah.
So I'll keep talking while Paul is working. But to me, it also shows something else in addition to how evolution works in bacteria. It looks like, if you just had to describe that without knowing anything about how it worked, you'd say, well, the bacteria want to, they want to move. They want to keep going. They want to occupy the whole dish. And what do they do to do that? They evolve. Okay? So when I talk about purpose in evolution, to me, that's an example. I mean, they could just sit there and wonder, why do they need to go into the other zones? There is a purpose that bacteria have to keep growing, keep dividing, increase the population size. And we know that, and it happens all the time. And often bacteria need to mutate, need to evolve to be able to do that. And that's one of the reasons that we certainly try to not take too much antibiotic, because if we do, we help the bacteria evolve antibiotic resistance. And that's another picture there. So let me see. Let me skip this slide. Wow, that really screwed everything up.
Yeah, video's hard.
All right.
Question.
Yes.
While he's doing that.
Good idea.
But the mutations that the bacteria undergo, aren't those random?
Yes.
Some are favorable,
Absolutely.
and some are not favorable.
Well, exactly. And that's a fascinating issue because it's not always necessarily the case that those mutations are new. They could have, what we really know about how evolution works is that right now in this room, there are many, many variants of every gene. Different people have different variants. Different animals have different variants. When the environment changes, it could favor one of those changes. So there could have been a bacterium already in that group that had a pre-existing mutated variation. And that mutation may have occurred a long time ago. And that mutation allowed the bacteria to live at a low concentration of antibiotics. So it was able to move over. But you're right, and that's why natural selection is so important, because if we just talk about the kinds of mutations, if you have 1 million bacteria and you get a few mutations, even if 90% of those mutations are harmful and they kill the bacteria, so what? You only need one mutation that's beneficial. And that's why you see colonies, because all you need is one organism, one cell that actually survives, and it will then grow and fill the plate. Now, that's a fairly simplistic view of evolution because it doesn't actually work exactly that way for larger animals, but it's kind of the same thing. If there are animals that have thick fur and not so thick fur, doesn't matter, they both hang around, and all of a sudden the weather gets colder, those with thick fur will do better. So natural selection literally selects what is the most beneficial genotype, the most beneficial mutation. Yes?
It seems to me like you're back to the turtle, though. Did they evolve so they could go forward, or did the ones that evolved correctly just then were able to go forward without necessarily doing it on purpose?
The second one. We don't know of any evidence that there's any mechanism for any creature to think about what should we do next. There's one exception, of course, which is us. We actually can change, and we obviously have, and we probably will continue to. But other creatures, no. There's no evidence or mechanism that would allow that. So natural selection is sort of this whole purpose thing is for biology is more of an unconscious, not willful kind of change, but it still functions as a purpose, and I'm going to get into that hopefully once we get the slides. I guess you got it. Yeah. Thank you, Paul. Yay. Okay. Now, I just hope I don't hit the back button. You don't want to go back to that. Okay. So I want to talk a little bit about, actually, that was a very good lead into this, which is where does the biochemistry come into all of this? And that's very important because, but that connection between the change in the genes, the genotype, okay, the genotype is all the genes, all the DNA sequences in an organism. The connection between that and evolution is not an easy thing because DNA doesn't do anything. It just sits in the nucleus. It's a book. It gets read, but just having a book doesn't do anything unless you open it and read it. And so making a mutation in the DNA by itself is of no use. It's what happens after that. And what happens after that is there is a very tight linkage, and everything I say now applies to every living organism we've ever seen, and also to what we presume to be the original first, what's called the last universal common ancestor, LUCA. LUCA is what we have deduced to be the first organism that all other organisms came from. It was a single cell, and everything I say applies to that also. Before LUCA, we have no idea. It's like the Big Bang. We can't go back any further right now. Maybe never. So what we have in all living organisms is a very tight linkage between the genotype and the phenotype. Why is that crucial? Because natural selection doesn't look at the DNA, it looks at the phenotype. The phenotype is all your characteristics. How fast are you? How thick is your hair? You know, how good is your eyesight? Those are phenotypic characteristics. Now, they come from the genotype. So there has to be a very tight linkage between that genotype and that phenotype. And that's where biochemistry comes in. The linkage is provided by the translation of the code, which is in DNA, into the set of proteins. All phenotypes are coming from proteins, whether they're enzymes, structural proteins, whatever. That's what gives the phenotype to a cell. Okay? Proteins are the things that make all the chemical reactions occur. They hold the cell together. They do all the really important stuff. Now, RNA and DNA are important, but only when it comes to the production of proteins. So that's the phenotype. And the tricky part is translating the nucleic acid-based genetic code into protein structures. And the reason I say that's tricky is because there's no common chemistry between amino acids or proteins and nucleic acids. So it's an incredibly difficult process to translate chemically one to the other. It's as if you had a foreign language and no Rosetta Stone. Okay? How do you translate one to another? Well, you could guess, but you're not going to get very far. So here's my hypothesis. Teleonomic, or purpose-driven, evolution derives from the biochemistry of translation. The process of protein synthesis, according to a genetic code, is goal-directed and the source of purpose in all of life. That's my hypothesis. I'm not going to prove it, but I'll give you some indications that might suggest it has something behind it. Okay, let's take a look. Evolution is actually a cycle, okay? Because what you have is you have a genotype, then you may have some mutations, then you have a new phenotype of a generation, then you have natural selection acting on that, the organism reproduces, and you have a new genotype, a new phenotype, and this goes on and on. And so what that is that, let's see if I have this here. No. The DNA makes the proteins. The protein is the definition of the phenotype. The environment acts on the phenotype, and natural selection acts on the interaction between the environment and the phenotype. Also, the natural selection decides which phenotype goes on to reproduce and do the new genotype. That's where natural selection comes in. And that's heredity. But the connection between the DNA genotype and the phenotype, which is absolutely critical in this cycle, is biochemical. And that's the biochemistry of translation. So what I want to talk about, this is going to be a little bit of a biology lecture. Feel free to pay no attention. I just love it so much that I can't resist talking. I used to be a professor, as Paul mentioned. So what has to happen here? It's DNA-directed protein synthesis. DNA makes protein. And as I said, you have to go from nucleic acid chemistry to amino acid chemistry. And lots of things are necessary. It's an incredibly complex process. You need something called messenger RNA, which has the codons. A codon is a piece of code that connects with one particular amino acid. You have to have transfer RNAs, tRNAs, which have something called an anticodon and various binding sites. And there's an enzyme, actually there are 20 enzymes, one for each amino acid, which has a terrible name. I wish somebody would change it to amino acid tRNA synthetase. And that enzyme is truly unbelievable. I'm going to show you more about it. It's my favorite enzyme in the world. And then there's a ribosome with lots of stuff and a lot of other stuff. But this is that enzyme. It's a protein, like all enzymes, and it has two binding sites. You see there are two different ones here. The red one has one kind of binding site on the left. The left binding sites bind a specific amino acid, and only that amino acid out of 20. That's kind of tricky because a lot of the amino acids look very similar. They're about the same size and shape. But these enzymes are able to only get the right one. That's really important. On the other side, on the right side, the same protein will bind something called the tRNA. The tRNA actually contains the code. And so the trick here is to take the right amino acid and stick it onto the adapter molecule, the tRNA, which has the right code, and then that tRNA will bring that to the messenger RNA, which came from the DNA, and put it in just the right spot. I know, it sounds crazy, and you probably didn't get any of that. Fine. You'll get it in a few minutes. We're going to keep hammering this. But that's one enzyme that does all of this, and it does a lot more. Once the two things are attached, and in this case, the red one, for example, has one amino acid and its correct tRNA, okay? Well, it has to be the right one in both. It puts them together. It makes a complex of the two. And now that complex goes into the ribosome and starts making protein, one part of the protein. Now, here are these tRNAs with their amino acids attached on the top. And notice, if you look down at those letters, the glycine, the code for glycine is CCC. Now, this is a little molecular biology. CCC will bind with GGG, which is coming from the DNA. So that is how, you don't have to listen to this. What you eventually get, though, is you get each amino acid in exactly the right position as specified by the DNA sequence. Okay? Anybody think this occurred by chance? It might have. I don't think so. Anyway, eventually you get an entire protein. But I want to say one more thing about my favorite enzyme, AAT synthase. I said that, sometimes the wrong amino acid will get in because, for example, some of them, valine and leucine, are almost the same. There's just one methyl group different. So how does it tell the difference? That's pretty chemically difficult. Well, it turns out that same enzyme has a special site called an editing site. And what it does is, if the one on the left is the correct amino acid, it will not fit into the editing site. It tries, it doesn't work, it goes back to the right place. But if you have one that's too small, it's a little bit too small, doesn't quite fit in the editing site, it doesn't quite, it's not the right amino acid, it will fit into the editing site. The editing site is slightly different. It will accept a lot of stuff, but it will not accept the correct amino acid. It will accept almost everything else. And so it stays there. And when it stays there, it gets degraded by the enzyme. Wow. There are people who are not as smart as this enzyme. This is amazing. And so the wrong amino acid gets thrown out. Okay. I say they have a purpose. Now, they're not thinking, okay? They're not saying, let's be careful we get the right amino acid, guys. Let's not make a mistake. No, they're not saying anything. This is a chemical. But it's a chemical with a purpose. Okay? It's not a conscious purpose, but it's just as conscious as the rabbit is to run away. Or maybe not a rabbit, but maybe it has a little more consciousness, but just as purposeful as those bacteria who are trying to spread out as much as possible. You can't not use teleological language to talk about this enzyme. It has a purpose to put the right amino acid on the right tRNA, and it does it beautifully. It binds the right amino acid and activates it. It binds several tRNAs based on their shape. It edits it to make sure it's correct, and it attaches the amino acid to tRNA. And every one of those 20 different ones has a different strategy. So each one is like its own, came up with its own ideas. But, you know, they're all different chemically. It's a remarkable thing. So, when you want to talk about how these enzymes work, and these are not philosophers or theists or whatever, these are just in textbooks. And the words in bold are what I call teleological language. As a consequence, enzymes facing this problem have evolved a second site. That's the editing site. These constraints have guided the evolution. They reach optimum ratios. And then the last question, I love it, how the synthetases deal with this. Well, we're talking about chemicals. We're not talking about, you know, a group of people sitting in a church meeting dealing with a problem. You know, this is a chemical, but it figured out a way to deal with it. Now, did this all arise by evolution? That's what most biologists will say. But I have a question that has no answer. This entire process is essential to evolution. How do you evolve something before there was evolution? I don't know the answer to that. There are some possible answers. When I've spoken about this with other biologists, they've said, well, it's a different kind of evolution. It's an error-prone evolution. The problem with that answer is, you know how good this translation process is? It's incredibly good. The fidelity, in other words, the number of mistakes, is 0.001%. And when DNA replicates itself, it's even better than that. It's 0.00001% of error. Now, we have to have some error, otherwise there would never be a mutation, and we need mutations for evolution. So there is some error, but it's incredibly low. And if you had a process that was error-prone for this translation, life not only wouldn't evolve, it wouldn't exist. Yes, sir?
So is your point that this process had to be available to this first organism?
Yes. Luca had this. There's no controversy that Luca had this system. Luca also could do photosynthesis. It could, well, it may not have been photosynthesis, but it had some energy. Either photosynthesis or what we do with you know, energy use. It had all the metabolic, major metabolic cycles. And it definitely had translation. So we do not know the origin of translation. The best guess for the origin of translation has been what's called RNA world, that before there were proteins, everything was done by RNA. That turns out probably not to work for technical reasons, which I'm not going to go into. But even that doesn't answer the question, because how did that become a DNA protein world? We don't know. Now, the fact that we don't know doesn't lead us to assume that there has to be something supernatural or divine about the process. That would be called God of the gaps. I don't want to do that. But I really don't have a good answer for how this whole thing could have evolved when it's absolutely essential for evolution. Now, it's not just my favorite enzyme that shows teleonomy. If you think about it, every enzyme is teleological in the sense that enzymes are catalysts, okay? We know what a catalyst is. It's something that does something, but it doesn't change itself. And there are many catalysts in the world. Catalytic converters are catalysts. And catalysts are important in chemistry. But biological catalyst enzymes are far, far better as catalysts than anything else. They're very specific. They only catalyze one reaction involving a single set of reactants. That's not true for any other, metal or any other clay, any other kind of catalyst. Those are usually very broad. They're incredibly efficient. The reaction rates are 10,000 times more than the basal rate. But one thing that people don't realize is that chemistry doesn't happen very much. Chemical reactions are very rare, and they're very slow. I remember when I first got my first chemistry set when I was a kid. My father was a chemist, so he gave me a chemistry set. And he said, be careful, just follow the instructions, which is something I have never learned to do. As you can see from my skill with the projector. So I just took all the chemicals and mixed them together. I figured the more chemicals you put in, the more likely to get an explosion, right? Nothing happened. Nothing. But very frustrating. But that's the real world. Chemical reactions occur. They occur in the atmosphere, they occur in the ocean. But they are random, and they are slow. And the kinetics are just ridiculous. Enzymes change all that. We could not live if we couldn't use really fast, I mean amazingly fast chemical reactions. And there are millions that go on every second in every cell of your body. And they have to go that fast. Otherwise, we would be really slow moving. There are incredible control and feedback mechanisms that enzymes are part of. And that's a whole field of biology. They deal with energy. Some reactions will not go because they're thermodynamically unstable or they're not favored. Enzymes take care of that. They will add another reaction to a reaction they really want to do, like make something, the synthetic reactions. Okay, you want to make a lipid to make a membrane. It just doesn't happen thermodynamically. You have to drive it with some other reaction, and enzymes do that. No other catalyst even could imagine that. And many of them, or most of them, are only produced when they're needed. And that's a whole nother very complex field of biology, of gene regulation. So enzymes are incredibly teleological. They certainly have purposes, and they're beautifully designed to do those purposes. So what is the source of all this purpose, this teleonomy in evolution? The existence of purposeful behavior in the current functioning. Now, I say current functioning because I don't want to talk about how it originated, because I don't know. But in the current functioning of this enzyme and other enzymes is undeniable. The history of its origin is unknown. Okay? That's as much as we can say scientifically. So there are two possibilities for that. It's part of the chemical, naturalistic abiogenesis of life by a more primitive form of evolution, and that form of evolution is unknown. We don't know how chemicals evolved to start life. There's no way to know that. Well, there may be one, but we don't know it yet. It's certainly not the evolution that we know exists in biology. I think I've proven that, or at least shown it. And of course, the second one is what I believe, that it's part of the creation of life by a creator. Now, that's also however, even though I believe it, it's also very tricky, because as a scientist, if that's true, I want to know how. And there has to be an answer to that. And I probably will never find out how in this life, but I would love to know that. So what my purpose here has been, not to convince you that there is divine or design or intelligent design in the origin of life, because I don't know if that's true, but to at least make it clear that we should never say that there's no teleology in biology. There clearly is at every level, in the behavior of animals, plants, and in the existence of the incredible biochemical processes that allow evolution to exist. And I think that's it. Thank you.